Hello, good evening to all of you. Here I am Murali PG, going to have classes under Unacademy regarding with the Karnataka CET 2021. We are focusing towards 2021 and here this is the information regarding with the plus subscription. So Unacademy is going to show the subscription regarding with the plus course. So plus course refers to best plans for Karnataka CET 2021 aspirants, and that comprises of 12-month plan at 12,375 and 6-month plan is about 10,125 and trial offer with respect to 1 month of about 999 and the price value till 15th of August. So in the plus subscription, what are the basic things? Here, premium content at affordable price, structured live courses plus dedicated live doubt clearing sessions, quizzes at regular intervals for self-analysis, batches for complete preparation, test series for DPPs, full-length mocks, etc. with analysis, single subscription for unlimited access. So if you go with the 12-month plan, you will going to get a single subscription for unlimited access. You can access the PCMB 4 subject with respect to all the educators coming under that PCMB. Okay. So if you want to subscribe for plus subscription, go to unacademy.com and there apply for plus subscription. Use my referral code MuraliPG853057 for the use and you will get a small reduction in that price. Okay. So you can get more at my plus course. Apply my referral code and use this link bit.ly/igetCET. So this link is for 2021 CET aspirants. Okay. So by this brief introduction with the plus subscription, now we will going to move with the next aspects. So this is the amount. This is the amount, that is Karnataka CET subscription. Here 1 month plan, 3 month plan, 6 month plan, 12 month plan. Okay. So see the difference for 6 month 10,125, 12 month 12,375. So see the difference among 6 month and 12 month. Okay. So welcome to all, welcome to the students, a warm welcome to Unacademy YouTube class, and here we are dealing with the MCQ question on biotechnology, its principle and processes. Okay, here we are studying biotechnology, its principle and processes. So questions under that chapter. So biotechnology is one of the very important chapter, and biotechnology is one of the important field with respect to job courses, and for this you have to achieve good in the oncoming exams. So Karnataka CET subscription is good under plus subscription of Unacademy. Okay, so let us have the beginning of discussion. So we will going to have the discussion of this class now. Okay, yes, so it is MCQ on biotechnology principle and processes. Myself, Murali PG, one of the biology educator in an academy. Then first question: the enzymes that cut specifically recognition site in the DNA is known as what? So in the case of recombinant DNA technology, we want the cutting of DNA in a specific recognition site, which is that enzyme: DNA ligase, DNA polymerase, reverse transcriptase, restriction endonuclease. So here the DNA, the enzyme that cut the specifically recognition site in the DNA. In the DNA, it is known as what? Specific site. It is the restriction endonuclease. Restriction endonuclease. Why restriction endonuclease? It will cut the DNA at a specific restricted area where palindromic sequences are there, and this is needed for our future use. That cut end portion is needed for our future use. So that restriction enzyme will going to work in a specific site. What about DNA ligase? Anyone can answer in the chat box. What about the DNA ligase? Anyone know? You don't know? So what is a DNA ligase? It is the important enzyme required for ligation, joining of two DNA molecule. Then it is called DNA ligase. Then it is called DNA ligase. Okay. Then polymerase enzyme. It is important. DNA polymerase is important to prepare another copy of DNA under transcription. Under a replication. Then reverse transcriptase. It is the enzyme that is required for the conversion of RNA into DNA. That is what reverse transcriptase. In the case of HIV, we can observe reverse transcriptase mechanism, especially in the viral RNA. It is working. Okay. So answer for the first question, it is restriction endonuclease. Next question number 2. DNA can be introduced into any cell by which method? DNA can be introduced into any cell by which method? Injection, being complexed with the calcium salt, gel electrophoresis, being placed along with the cell. Which is the right answer? So here DNA can be injected or introduced into any cell by injection, means microinjection, being complex with calcium salt, gel electrophoresis, being placed along with the cell. So it is answer is this one, A, microinjection. So specialized needles are there. By using that one, we can directly introduce the DNA of our interest into the host or into any cell. So answer for this question, A is the right answer for second question. Okay. Next, third question. Ability of if you have any doubt, you can give the questions, or you can ask in the chat box. Okay. So third question. Ability of a plant or animal cell to repeatedly divide and differentiate into a complete organism. Ability of a plant or animal cell to repeatedly divide and differentiate into a complete organism is what? Cloning, DNA fingerprinting, cellular totipotency, mitosis. Anyone can answer in the comment box if you know the answer. So here it is what? Cellular totipotency. So cell which shows totipotency, which shows the ability to give rise to the entire organism, then it is called totipotency. Here in the case of tissue culture, the explants will going to show the totipotency. So answer for the third question is C. Answer for the third question, it is C. Next question, number 4. Yes. Restriction endonuclease is also known as what? Molecular glue, DNA ligase, DNA polymerase, molecular scissors. So restriction enzymes will work on DNA molecule. So they are actually what? Molecular scissors. They will work against DNA molecule. They work against molecular level of biological system. So molecular scissor is the right answer. Answer is D for question number 4. Molecular scissor. Okay. Next question, number 5. Yes. Okay. Next question, number 5. Yes, it's coming. Yeah. Extra chromosomal small circular double-stranded DNA molecule in a bacterial cell is known as what? Extra chromosomal small circular double-stranded DNA molecule in a bacterial cell is known as what? Plastid, Plasmid, Mitochondrion and Chloroplast. So which is the right answer? So for the 5th question, B is the right answer. B is the right answer, that is the Plasmid. Okay. Introduction of foreign genes into plant or animal cells using micropipette is what? You see, introduction of foreign genes into plant or animal cells using micropipette. So micropipettes are going to be utilized for the introduction of foreign gene into plant cell. Which is that one? It is called chemical-mediated gene transfer. Chemical-mediated gene transfer. It is not electroporation. It is not microinjection. It is not particle gun or gene gun. So gene gun is different. and microinjection direct introduction of genetic material through specialized injections. Okay, needle. By using specialized needle, specialized needle, we can observe the introduction of the foreign gene. Then electroporation is electrical charge should be applied, but here it is a chemical mediated gene transfer, that is what, using micropipette. So answer is A for 6th, answer is B for 6th one. And next, the 7th one. Yes, 7th one, whether it is visible for you. Yes. Which one of the following is related with the genetic engineering? Mutation, ribosomes, mitochondria, plasmid. Which is the more related word related with the more often using word, that is called plasmid, isn't it? Plasmids are the vectors which will carry the gene of our interest into host body, host cell, to produce the product or expression of character. So plasmid is the answer for question number 7. Next, 8th one. What it? Next, the 8th one. Yes. In bacteria, yes, okay, yeah. In bacteria, genes for antibiotic resistance are usually located in which area? Plasmid, cytoplasm, mitochondria, nucleus. In bacteria, genes for antibiotic resistance are usually located in cytoplasm, because the pure nucleus is not there in the case of bacteria, isn't it? It is called nucleoid. So genetic material is present in the nucleus. So cytoplasm, genetic material is present in nucleus in the eukaryotic cell. In the prokaryotic cell, it is present in the case of cytoplasm. There is no separation between the cytoplasm and inner genetic material, nucleus genetic material. Nuclear membrane is absent in the prokaryotic cell, for example bacteria. So in bacteria, genes for antibiotic resistance are usually located in cytoplasm. So the answer is B. Okay. Next question, question number 9. A technique used to make numerous copies of a specific segment of DNA quickly and accurately. So making of number of copies, but it should be specific segment of the DNA. Quickly and accuracy should be there. Which is that method? Translation. Translation is what? It is a process of formation of protein. Transcription, it is the production of mRNA. Ligase chain reaction, it is a ligation phenomenon, joining. What about polymerase chain reaction? So polymerase chain reaction is the right answer for this technique. So which is used to make numerous copies of specific segment of DNA quickly and accurately. So accuracy is there and quick nature is also there. So it is easy to handle. So here we are getting a clean setup with respect to this entire DNA sequence. So small set of DNA, small stretch of DNA can going to be amplified by using polymerase chain reaction. So answer is 9. For 9th question, answer is D. Okay. Next question. The enzyme that cleaves DNA at specific sites producing sticky end is called what? So got it? The next question number 10. Banidala? Yeah. The enzyme that cleaves DNA at a specific site producing sticky end is called what? So what is that enzyme? Exonuclease, lysing enzymes, cleaving enzyme, and restriction endonuclease. The answer is restriction endonuclease. If you have any doubt, you can answer. You can ask. Okay. If you know the answer, you can answer in the chat box. You can give the answer in the chat box. Okay. So answer for 10th one, A is the answer. Okay. Next question number bantala. Yes, which of the following is a genetic vector? Genetic vector. Plasmid, phage, cosmid, all of these. So which is the most probable use of genetic vector, or in most of the case we are using that one as a genetic vector? Which one? That is the plasmid. That is the plasmid. If it is phagemid, then we can consider this one of the genetic vector. But it is not phagemid, it is a phage. So plasmid is a clean genetic vector. So answer for 11th question, A is the right answer. Then 12th question. Restriction endonuclease are used in genetic engineering because they can degrade harmful proteins, they can join DNA fragments, they can cut DNA at specific base sequences, they can cut DNA at variable site. So 12th question, let us have the answer now. So they can cut or they can degrade harmful proteins. Agta? No. They can join DNA fragments. That's a ligase enzyme, isn't it? They can cut DNA at specific base sequence. They can cut DNA at variable site. So right answer, correct answer is C. Alva. So they can cut DNA at specific base sequence. So answer is C for question number 12. Because of that reason, it is called restriction enzyme. Next, 13th question. What it? 13th question. Ideal host for amplification of DNA molecule is amplification of DNA molecule can be done by using following host, which is the right answer: virus, plants, bacteria, animals. So 13th one, anyone can answer in the chat box. It is what is that? So 13th question, it is C, isn't it? Bacteria. Bacteria is the right answer. So it is ideal host for E. coli cells, ideal host for all the reactions. So who given that answer? Yes, KSMS, MKSM. Yeah, you are right. Next question number 14. TA plasmid naturally occurs in which bacteria? Agrobacterium, cyanobacteria, staphylococcus, and vibrio. So TA plasmid naturally occurs in what? Agrobacterium, tumefacian, Agrobacterium, tumefacian, Corynebacterium, staphylococcus, and vibrio bacteria. So here The answer is okay, Prekshita. Okay. So answer is what? Agrobacterium. So A is the right answer, Prekshita. You are right. Good work. So what will going to happen? This Agrobacterium tumefaciens will going to cause crown gall disease. Is it a crown gall disease in the plants? So TI plasmid, in that tumor inducing cells are present, or nature is present. Okay, so question number, where we are, yes, yeah, 14th question alva, yes. So it is Agrobacterium tumefaciens. Next question number 15. The sticky end of fragmented DNA molecules are made up of what? Sticky end. During the restriction endonuclease action, the sticky ends are going to be formed. That sticky end is made up of what? Which molecule? Prekshita, you know the answer? So here the answer is for 15th question, it is D, methyl group. Methyl group is A, okay. Which of the following are the essential requirement for recombination? Essential requirement for recombination, or in the recombinant DNA technology, what is the essential requirement? Single stranded DNA, DNA ligase, DNA polymerase, all of the above. So 16th one. So which is the answer? Do you know, Prekshita, for question number 16? It is not all of the above, it is DNA ligase. DNA ligase is very important for the fusion, essential requirement for recombination. After cutting off unwanted DNA, in that place we are inserting our interested DNA. So for that, this DNA ligase is very important. Okay. So answer is B for question number 16. Next 17. Another question? Yeah. The plasmid derived from E. coli is which one? PBR 327, PBR 322, both the above, both A and B, and none. So which is the right answer? Here for 17th question, plasmid derived from E. coli. It is called what? So PBR 322. Is there any other answers? Is there any other answers? No. So actually the answer is B, PBR 322. Okay, not PBR 327, 322. Then TI plasmid is useful in what? TI plasmid is useful in what? Bringing new genes into animal cell, bringing new genes into plant cell, to nearly any site on a chromosome, bringing tumor cells into plant cell. Which is the right one? So remember, TI plasmid is useful in what? Any answer? So Ti plasmid will going to have the tumor inducing property. So in this one it is bringing tumor cell into plant cell, isn't it? Plant cell, tumor cell into plant cell, that is one type of answer. Then here bringing new genes into plant cell, that is also right. Actually this is the answer. B is the right answer, because in the point of if you are analyzing with respect to human benefit, it is bringing a new gene into plant cell using this Ti plasmid. We can introduce the required gene into Ti plasmid of Agrobacterium tumefaciens and introduction of that one into plant cell. So as a result of that, new gene will arise, or it will going to reach. Here bringing tumor cells into plant cell as a general function. Regarding with the biotechnological aspects, principle and process, this is the right answer. That is B is the right answer. That is Prekshita. Okay. So answer is 18th question. Answer is B. The 19th question. Many copies of DNA molecules in a test tube are procured by which method? Many copies of DNA molecules in a test tube are procured by. So it is how we can get many copies. Many copies. Rakshita, how many copies of DNA molecule in a test tube are procured by which method? That is the polymerase chain reaction, PCR, where we can amplify the DNA molecule. So answer is A for yes, A for the question number 19. Then 20. What is this? BAM H1C is echo R1 Sal 1 are the types of what? So what is that thing? BAM H1, echo R1, Sal 1 are the type of what? It is a type of, do you know? Answer, students. So answer for this one, for 20th question, it is A, restriction endonuclease. Okay. So answer for the 20th one, A is the answer. Next question number 21. Retroviruses have genetic material which is made up of what? Retroviruses with genetic material made up of what? Shama, Prekshita. It is RNA. It is RNA, because RNA is having RNA virus or DNA as genetic material. So it is called RNA virus and DNA virus. Virus may be RNA, virus may be DNA. Virus may be having DNA. Retroviruses have genetic material in which RNA is the major component. First it is made up of RNA. Next question number 22nd. Got it? Yeah, genetic engineering is possible because the phenomenon of transduction in bacteria is well understood. We can see DNA by electron microscope. We can cut DNA at a specific site by endonuclease like DNase. One distinction endonucleases purified from bacteria can be used in vitro. So genetic engineering is possible because of what? Answer is there for this 22nd question. Answer there. So here the answer for 22nd, it is A. The phenomena of transduction in bacteria is well understood. So this is the right answer. Transduction in bacteria very understood because the genetic material of bacteria is very small, easy to handle, easy to study, isn't it? So the entire phenomena of bacteria regarding the genetic material that we already studied in the research work, isn't it? The human being scientist. Then plasmids are the suitable vectors for genetic cloning as what? Why it is suitable vectors? That is 23. So answer for 23, it is they are self-replicating unit. B is the answer. Any plasmid, they should have origin of replication. Then only it is good for the research work. After entering into the host body, the plasmid should have the capacity to undergo self-replication. Okay, so answer for 23rd, it is B. Prakshita, you given B. Okay, good. Now 24th, which of the following is used in genetic engineering? Restriction endonuclease, mycobacterium, entamoeba, pepsin. So which is the right answer? Which of the following is used in genetic engineering? 24, it is answer is which one? Prakshita, it is the restriction endonuclease. So answer is A, restriction endonuclease. Yeah. Next, the first hormone artificially produced by culturing bacteria is which one? The first hormone artificially produced by culturing bacteria is which one? Insulin, thyroxine, testosterone, adrenaline. So 25th question, the answer is what? Do you know? It is Ren. Yes, answer is insulin. So first hormone artificially produced by culturing bacteria is insulin. Insulin is going to be produced by Eli Lilly company. The first artificial human insulin is called humulin. It is called humulin. Okay, humulin. So answer is A for 25th question. Prakshita, you are right. Yeah. Next question, number 26. When the number of genes increases in response to some signal, the effect is called what? When the number of genes increases in response to some signal from the outer side, the effect is called what? 26. It is called what? So increase in the number of genes, that is what? Amplification. Yes, Prakshita, you are right. Gene amplification. Okay, So we are going to get a large number of genes of a small size. So from a small size we can amplify that gene. So we can get a large number. Answer is C for 26. Then 27. Which one of the following pairs is correctly matched? Correctly matching pair, which is the right thing. Correctly matching word. RNA polymerase, RNA primer. Then restriction enzymes, genetic engineering. Central dogma codon. Auxiliary fragment splicing. That is correctly matched. Which one of the following pair is correctly matched? So answer is for 27. Do you know? It is B. Restriction enzyme, genetic engineering, isn't it? Restriction enzyme is going to be utilized for genetic engineering purpose. RNA polymerase is for the production of RNA. Central dogma codon Allah. Central dogma says that from DNA it will undergo self-replication to produce another copy of DNA. DNA will produce mRNA by transcription. mRNA will undergo translation to produce protein. That is central dogma. Then auxiliary fragments are codon refers to triplet codon, codes for one amino acid. Auxiliary fragments refers to these are the fragments of DNA which is formed in the lagging strand when the DNA replication is going to be happening. Later these auxiliary fragments are joined by using DNA ligase. Then splicing is the process where matured mRNA is synthesized in eukaryotic transcription. So it is having both intron and exon, where introns are not in function. So introns are removed, exons are united. For that splicing phenomena, spliceosomes will be appear. Spliceosomes will be there. So that is one. They are incorrectly matched. So correctly matched is B. Okay. Answer is B for 27th. Next question number 28. Sorry, 28. Plasmids are autonomously replicating mini chromosomes found in where? So plasmids are autonomously replicating mini chromosomes found in where? Yes. Any answer? 28. It is Escherichia coli. It is the Escherichia coli. Okay. It is the Escherichia coli. E. coli cells. Autonomously replicating mini chromosomes are found in Escherichia coli, one of the important organism under this biotechnological industry. So answer for this question, it is C is the right answer. Next question number 29. Yeah. Improvement of genotype of an organism by the addition of some foreign gene is called what? Improvement of genotype of organism. You can improve that genotype of organism by what method? By addition of some foreign gene. That phenomena is called what? 29. Answer is genetic engineering. Genetic engineering. Akshita? Yeah. Genetic engineering. Next 30th question. Two bacteria found to be very useful in genetic engineering experiment. Which are the two bacteria? In a genetic engineering experiment, these two bacteria are very much important, have significant criteria, have good positive role in the genetic experiment. Which are the bacteria? Yes, Prekshita, you are right. That is B is the answer. Escherichia coli and Agrobacterium tumefacium. Yes, next question, number 31. Restriction enzymes. Restriction enzymes are isolated chiefly from which group of organisms? Algae, fungi, protozoa, prokaryotes. From which one we can observe highest isolation of restriction enzyme? So chief main isolation is from which group of organism? It is prokaryotes. Okay, it is prokaryotes, not protozoa. It is prokaryote, bacteria, etc. It's not protozoa. Protozoa means amoeba. They are the eukaryotic organism, unicellular eukaryotic organism. So answer is D, prokaryotes. Then there are special protein that help to open up DNA double helix in front of the replication work. These proteins are what? See, special proteins that help to open up DNA double helix in front of the replication work. So during the replication work, this particular protein will going to help in the unwinding of DNA helix. The protein is what? 32nd question. So the answer is DNA guidase. Unwinding of DNA helix is done by DNA guidase. So as a result of that, unwinding of DNA molecule, reading of that molecule, and replication can be completed. So answer is A for question number 32. Then 33 question. Yes, got it? Yes. Technology which uses living components for the welfare of human being is what? Biology, botany, bioinformatics, biotechnology. Technology which uses living organisms or living components for the welfare of human being. So under this, which is the right answer? Anup, welcome. Yes, biotechnology. Biotechnology is the right answer for, biotechnology is the right answer for question number 33. Okay. Yes. Prekshita also, you are right. Okay. Then, which process is involved in making bread, cheese, beer and wine? Which process is going to be there? Respiration, hydrolysis, degradation, fermentation, decomposition. Difficult questions are up there. Already it is that questions were passed now. Yeah. Some of the questions are difficult, some are moderate, some are easy. That is the type of CET paper. Okay. So next question. So here 34th question answer, it is C, fermentation, isn't it? Fermentation. What is fermentation, Anoop? What is fermentation, Anoop? Okay, I will continue. Okay. So EFB stands for what? EFB stands for what? European Foundation of Biotechnology, European Foundation of Biology, European Foundation of Biotechnology, European Foundation of Biology. So here twice the same options, the yes or no, which is the right one. Okay, you can take any one out of B and D and any one out of A and C. So what is that answer? 35. It is C. European Foundation of Biotechnology refers to European Foundation of Biotechnology. It is one of the biotechnology related foundation. Okay. Next question number 36. 36 question. Yes. The organism whose gene has been artificially altered for desired effect is called what? So organism whose gene has been artificially altered for desired effect is called as what? Genetically mutant organism, gene transfer, genetically modified organism, genetically transferred organism. Which is the right answer? The organism whose gene has been artificially altered for the desired effect is called what? 36 question. It is C. Genetically modified organism. Okay. GM organism, GM plants, GM animals are there. From that we are obtaining GM food. Okay. Yes. 37. The sequence of DNA that reads the same backward and forward across the double strand is called what? Recipient sequence, palindromic sequence, replicate sequence, origin sequence. So which is the right answer? The sequence of DNA that reads the same backward and forward across the double strand thing, that is called palindromic sequence. Answer is B. Okay. Next question number 38. How many restriction enzymes are known to be isolated? How many restriction? Yes, Rakshita, you are right. Nasima, you are right. So how many restriction enzymes are known to be isolated? More than 800, more than 700, more than 600, more than 900. So it is more than 600. So answer for this question number 38, it is C is the answer. More than 600 types of restriction enzymes were isolated in about nearly 275 prokaryotic cells of bacteria. Means 275 bacteria. Okay. So that is the capacity of the E. coli. Okay. Yes, Anup given 700, but it is 600. Okay. This Prakshita and Nasima, you are right. I am explaining whenever it is necessary. Okay. Which of the following step is necessary part of DNA recombination technology? So which are the steps important with respect to part of recombinant DNA technology? Insertion of DNA fragment into vector, insertion of vector into bacteria, multiplication of clones containing the recombination molecule, all the above. 39 question, let us see. So insertion of DNA fragment into vector, part of DNA recombination technology. Insertion of vector into bacteria means host cell. Multiplication of clones containing the recombinant molecule. So 39 answer is all the above. This is the process of DNA recombinant technology. So here we are cutting the unwanted DNA sequence of vector. In that place we are inserting our DNA sequence, and later it was introduced into a insertion of that vector containing a DNA sequence into a bacterial cell, and multiplication of the clones containing the recombination molecule in a culture media, and we are going to get the product, or multiplication will be there. So all of the above is the right answer. It's a brief step of the DNA recombinant DNA technology. So before doing that, isolation of DNA, then purification of DNA, all those things are there. So the answer is D for 39. Restriction enzymes belonging to which class of enzymes? Restriction enzymes are made up or coming under which group of enzymes? Restriction enzymes coming under which group of enzymes? Nucleolase, exonuclease, nucleases, endonucleases. So answer is for 40th question, it is C, nucleases. In that we can bifurcate. One is restriction endonuclease and another one restriction exonuclease. Okay. Generally it is a nuclease. Nucleases are the enzymes, group of enzymes which will act on nucleic acid. So answer is C for 40th question. Next, 41st. A sequence of in a genome at which replication is initiated. Initiation of replication is observed in which area? So the answer for this one, 41. Got it? Yeah, 41. Yeah. A sequence of in a genome at which replication is initiated. In origin of replication, selectable marker, cloning site, origin of restriction. It is origin of replication. So origin of replication is the actual site where replication is going to start. So all the vectors should have ori site. Then only, that is one of the important character of vector, having one ori site. Yes, Guru Prasad, you are right. Next question, question number 42. Genes which help in the growth of transformants are what? Origin of replication, cloning site, origin of restriction, selectable marker. So genes which help in growth of transformants, which is that one? Cloning site, origin of restriction, selectable marker. So growth of transformants, you know, that is what? Selectable marker, isn't it? Selectable marker. So it is important to differentiate or isolate the transformant from non-transformant, isn't it? So selectable marker. So for 42nd question, The answer is selectable marker. These questions are from NCERT. You can refer, Anita. Okay, NCERT is common with all states. You can refer. Okay, for Maharashtra CET you can refer, because here these questions are from our NCERT textbook. We are focusing towards Karnataka CET 2021 because 2020 CET already over. Okay, you can use this one. You can have the classes regarding the Unacademy. In the end I will show the slide. Okay, yeah, you should be there, yeah, after discussion of all the questions. Another 2 or 3 questions are there. So selectable marker, that is 42nd. 44. TI plasmid is a cloning vector which works with the what? Tumor inducing plasmid. All plants dicots only, monocots only, thallophytes only. It is dicot only. Only with respect to dicot, where secondary growth is there, where they will going to form the crown gall disease. Okay, so if you want to introduce any gene into our plant through this Agrobacterium tumefaciens, we can have this the use of this TI plasmid of Agrobacterium tumefaciens. ಯಾವುದೇ ಪ್ಲಾಂಟಿಗೆ ಆ್ಯಡ್ ಮಾಡೋದಿದೆ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಎನಿ ಪ್ಲಾಂಟ್ ಆಫ್ ಡೈಕಾಟ್ ಟೈಪ್, you can use this TI plasmid. So answer for 43rd is B is the answer. And 44. During which of the following techniques host cells are exposed to pulse of high voltage current? During which of the following technique, the technical aspects of biotechnology, host cells are exposed to pulse of high voltage current? Electroporation, particle bombardment, microinjection, liposuction. So 44th question, it is electroporation. It's one method where introduction of gene. Electroporation. So high host cell is exposed with the high pulse of high voltage current. That is electroporation, not particle bombardment. Microinjection, liposuction is a gene gun. Microinjection, liposuction, but here we can use the use of high voltage current in the experiment, in that technique to deliberate the gene. So 44A. 45. Particle bombardment technique is also known as what? Just now I told, isn't it? Liposuction, electroporation, biolistic, microinjection. It is nothing but, yes, it is nothing but biolistic gun, particle bombardment or a gene gun. Why it is particle bombardment? There the coating of gold and tungsten particle will be there to the gene, and by using that one we are introducing the gene of our interest to plant cell. That is the particle bombardment or use of biolistic gun, gene gun, another term gene gun. Microinjection, that is a regular practice where a very small needle is going to be utilized. Okay, so 45th question, answer is C. So I think all the questions are over now. So now if you want, you can have this plus subscription under this Unacademy. So in the beginning I will show, yeah, Unacademy plus subscription, best plan for Karnataka CET 2021 aspirants. Even Maharashtra students can also have this one. So plus subscription. So best plans for Karnataka CET 2021. I think one of the student, Anita, Yeah. And 12-month plan, that is 12,375. 6-month plan 10,125, and 1-month plan 999, and till 15th of August the price is like this. Later it will be changing, and in this one 1 month or 12 month or 6 month, you will going to have a complete detailed syllabus of that particular chapter, along with MCQ, weekly quiz, doubt clearing sessions, etc, etc. So if you go with the 12-month plan of this much value, you will going to get unlimited access to all the educators PCMB and the educators PCMB subject with all papers of educators. Okay, then use code, my use code is Murali PG 853057. so see the difference of value 6 month and 12 month. So there will be 50% reduction in the 12-month plan. Okay, more than 50% if you are calculating. Okay, so that is one slide. This is the actual price value. Here it is given 54% off, here 25% off, here 17% off, here 10% off, like that. Okay, so if you are heading towards this year CET in some of the state, then you can have 1 month plan. Okay, along with that you will going to get the special classes, that is for free. Okay, special class are there, that is for free. There are also MCQ in the special class, only MCQ, but in the case of plus course detailed theory will be there. Okay, by this I am going to end up this session. Thank you one and all. So we will meet in the next oncoming YouTube class. Okay, thank you.